In May 2026, a Nature paper described something that had never happened before. An AI system called Robin, built by a nonprofit called Future House, had autonomously proposed a drug candidate for a leading cause of blindness, designed the experiments to test it, and interpreted the results. Humans only touched the pipettes. The drug, ripasudil, a glaucoma medication already approved in Japan, appeared to work. But buried in the supplementary materials was a second number. Robin's analysis agent, Finch, reported that ripasudil boosted a critical cell function 7.5 times over the control. When human scientists reanalyzed the exact same flow cytometry data, the number was 1.75 times, a fourfold gap between the machine's answer and reality. The drug was real, the effect was real, but the AI's measurement of its own discovery was not. For decades, the bottleneck in drug discovery has been finding candidates worth testing. It is a brutally slow and expensive process. The FDA approves roughly 50 novel drugs per year. 9 out of 10 candidates fail after human trials begin, often after billions of dollars have already been spent. The promise of AI in science has always been a simple one. What if we could generate hypotheses faster than any human team? In May and July of 2026, 3 separate AI scientist systems published in Nature and Science within 2 months of each other. Future House's Robin, Google DeepMind's co-scientist, and Stanford's Biomni. The hypothesis generation bottleneck is dissolving, but the Robin paper reveals a new bottleneck forming right behind it. If the AI produces hypotheses faster than humans can verify them, and if the AI's own verification cannot be trusted, then the speed of discovery is no longer limited by imagination. It is limited by the speed at which humans can check the AI's work. Robin is not one model. It is a system of specialized language model agents, each assigned a specific cognitive task, sitting on top of commercial foundation models. Think of it less like a single brain and more like a research team, except every member of that team is an AI. Crow runs fast, concise literature searches, scanning hundreds of thousands of open-source papers, patents, and clinical trial records in a fraction of the time it would take a human postdoc to read even a handful of them. Falcon runs deep, extended technical analyses on specific topics, generating the kind of granular molecular reasoning that would normally require months of domain expertise. Finch handles raw experimental data, RNA sequencing, flow cytometry, statistical interpretation, turning messy lab output into quantitative conclusions. And a tournament judge ranks hypotheses using Bradley-Terry paired comparisons, the same mathematical framework that sits behind chess ELO ratings. Every candidate hypothesis competes head-to-head against every other, and the system surfaces the ones with the strongest combined evidence. The workflow is iterative. Future House pointed Robin at dry age-related macular degeneration, known as DAMD, a disease affecting roughly 200 million people worldwide with no approved treatments. Robin read the literature. Crow proposed that enhancing phagocytosis, the process by which retinal cells clear debris, could slow degeneration. Falcon evaluated candidate molecules. Humans ran the bench experiment. Finch analyzed the data. The first round identified a ROCK inhibitor called Y27632 that showed promise. Robin then proposed an RNA sequencing experiment to understand the mechanism, which revealed that Y27632 was upregulating ABCA1, a lipid efflux pump critical to retinal cell health. From that insight, Robin proposed a second round of candidates. Riposidil emerged as the top hit, a drug already used in human eyes for glaucoma, never before proposed for DAMD. The entire process, from planning to manuscript submission, took 2.5 months. Future House estimates it cut researcher time by roughly 200 times over conventional methods. All hypotheses, experimental plans, data analyses, and data figures in the main text of the paper were produced by Robin, not assisted by, produced by. Start with what Robin got right, because it got something genuinely right. Riposidil enhanced phagocytosis in retinal pigment epithelial cells. The drug is already approved for ocular use in Japan. It has a known safety profile in human eyes. Doctors already prescribe it. ROCK inhibitors had been studied for wet AMD and other retinal diseases involving abnormal blood vessel growth, but nobody had proposed their application in dry AMD specifically for their effect on phagocytosis. Robin was the first. It saw a connection in the literature that human researchers, despite decades of accumulated knowledge about both ROCK inhibitors and macular degeneration, had not made. Robin also proposed a second novel mechanism, modulating the cell's circadian rhythm using an experimental compound called KL001 that had never previously been suggested for AMD in any form. Two original therapeutic hypotheses from one AI system in a single research cycle. This is not a simulation. This is not a benchmark score on a leaderboard. A multi-agent AI system read the scientific literature, identified a therapeutic strategy, named a specific drug, designed the experiments to test it, and the drug worked in the lab. Real cells, real effect, real biology. The preprint went up in May 2025. Nature published it 14 months later. Now consider what Robin got wrong and why the error matters more than the discovery itself. Finch, the data analysis agent, processed the flow cytometry results from the riposidil experiment and reported a 7.5-fold increase in phagocytosis over the DMSO control. That is a massive effect, the kind of number that makes a drug candidate leap to the top of any priority list. But the paper itself records that human reanalysis of the same raw data produced a 1.75-fold increase. The paper attributes the difference to a difference in analytical methodology, but a 4-fold gap is not a rounding error. It is the difference between a compound that changes the field and a compound that is mildly interesting. The drug still works. 1.75 times is a real signal, but the AI's confidence in its own result was structurally inflated. This matters because the entire architecture of autonomous science depends on the AI accurately assessing what it found. If the system that generates hypotheses also overstates the evidence for those hypotheses, then every downstream decision, which candidates to advance, which to discard, where to allocate precious lab time and funding, is distorted. A drug that looks 7.5 times better than the control gets fast-tracked. A drug that looks 1.75 times better gets added to a long list. Same drug, same data, different future depending on who analyzed it. The verification paradox is not a bug in Robin. It is not something that a software patch will fix. It is a structural feature of any system where the thing doing the discovering is also the thing measuring the discovery. When the judge and the contestant are the same entity, the score cannot be trusted at face value. Robin did not publish alone. The same week in May 2026, Google DeepMind's co-scientist also reached Nature. Co-scientist uses a different architecture, a tournament evolution system, where hypothesis agents generate candidates and reflection agents critique them using ELO-style rankings. Where Robin acts like a research team with distinct specialists, co-scientist acts more like a debate society. Hypothesis agents propose ideas, and reflection agents tear them apart. Only the ideas that survive repeated critique advance to the next round. DeepMind pointed it at acute myeloid leukemia, one of the most aggressive blood cancers, a disease where patients desperately need new treatment options. It proposed 30 drug repurposing candidates from a curated list of 2,300 approved drugs across 34 cancer types. Human oncologists reviewed the list and selected 5 for lab testing. 3 showed positive results. Binimetinib, already approved for melanoma, killed AML cells at nanomolar concentrations, meaning it worked at extraordinarily small doses. But an unnamed cancer researcher told Chemical and Engineering News that the work had not identified especially novel targets, and co-scientists' predictions were never compared against the decades of existing computational drug repurposing tools that have quietly produced similar lists for years. The question is not just whether these AI systems produce good results. The question is whether they produce results that justify their cost and complexity over what already exists. 2 months later, Stanford's BioOmni reached Science. Its preprint had been on archive for 13 months. Robin's preprint had been up for 14. The journal clock, the peer review process that has governed scientific publishing for generations, runs on a timeline that no longer matches the speed of the field it is supposed to certify. By the time Nature published Robin, Future House had already spun out a for-profit company called Edison Scientific, which raised $70 million at a $250 million valuation. Investors did not wait for peer review. They read the preprint and wrote the check. And Future House had already launched Cosmos, Robin's successor, before the ink on the Nature acceptance letter was dry. The published record is behind, the money is ahead, and the verification question sits unanswered between them. Every result described here is preclinical and in vitro. That distinction matters enormously and is easy to lose in the excitement. No AI-proposed drug candidate from any of these systems has entered human clinical trials, not one. Ripasudal enhanced phagocytosis in cultured retinal cells, not in a living human eye with all the complexity of blood flow and immune response and aging tissue that a dish of cells cannot replicate. Binimetinib killed AML cells in a dish, and it had already failed a Phase 2 clinical trial in AML before co-scientists ever proposed it. The drug had already been tested in actual leukemia patients and had not worked well enough to proceed. The gap between a cell line result and a working therapy is vast and unforgiving. It is where most drugs go to die. Roughly 90% of candidates that enter clinical trials never reach patients. And there is a deeper limitation the coverage rarely surfaces. None of these systems were benchmarked against the targeted computational biology tools that have existed for decades. Molecular docking software, network pharmacology platforms, quantitative structure-activity relationship models. These are tools that pharmaceutical companies have used for years to generate repurposing candidates. We do not actually know whether Robin or co-scientist outperform these existing, less expensive methods. We know the AI systems work. We do not know they work better. Robin found a real drug for a real disease. That is not hype. It is a genuine result that will matter to the 200 million people losing their vision to dry, age-related macular degeneration. But the same system that found the drug also reported an effect size 4 times larger than reality. Future House disclosed this honestly in the paper, in the supplementary materials. That honesty is the most important data point in the entire field right now. Because as AI scientist systems multiply, Robin, co-scientist, Biomni, Cosmos, and whatever comes next month, the question is no longer whether AI can generate good hypotheses. It clearly can. The question is whether anyone is building the verification infrastructure fast enough to keep up. The speed of discovery is now the speed of checking, and right now, checking still requires a human looking at the same data the AI already looked at and getting a different answer. Robin proposed one drug for one disease. It took 2.5 months and required human scientists to run every physical experiment. But Future House's next system, Cosmos, is designed to run hundreds of disease hypotheses simultaneously without human guidance at each step. The ambition is staggering. The infrastructure to verify it does not yet exist. What happens when an AI scientist that cannot accurately measure its own results is told to work on 100 diseases at once? The architecture scales beautifully. The verification does not scale at all. And somewhere between those two facts, between the speed of the machine and the patience of the human checking its work, sits the future of how we discover medicine.